Hello everyone and welcome back to our channel Quaint. Today we are diving into one of the most mind-bending ideas at the intersection of biology and physics. How could CRISPR and DNA technology might actually help us unlock the secrets of neutrinos? So DNA technology and CRISPR, these belong to what? These belong to biology, and neutrinos belong to physics. That's why I have said that at the intersection of biology and physics. Anyway, so you know the importance of neutrinos. You know, neutrinos are also called ghost particles because they rarely interact with matter. And for this reason, they are also called ghost particles of the cosmos. And they come from various sources. They come from nuclear interactions, nuclear reactions in stars, then exploding supernovae, then the Big Bang itself, and they carry information about the deepest secrets of the cosmos. They carry information, they means here neutrinos. Neutrinos carry information about the deepest secrets of the cosmos. So unlocking them could reveal, unlocking their secrets, unlocking the secrets of neutrinos could reveal how galaxies formed, why matter exists, and even some clues about the dark matter. You know dark matter, anyway. So that's why neutrinos are so much important. Means unlocking the secrets of neutrinos are so much important. And you know that right now, trillions of neutrinos are passing through you. Yes, right now, trillions of neutrinos are passing through you, but you don't even feel a thing. And it's true. And normally, various detecting methods are there. Actually, neutrinos are known as notorious. Actually, they are called notorious for their that behavior. Which behavior? Very hard to detect. Neutrinos are very hard to detect, and that's why they are called notorious. Okay, this nature is called notorious. They don't interact with matter properly. Better to say they rarely interact with matter, and that's why very hard to detect. But detection is necessary to know the secrets of neutrinos. First of all, what is the requirement? First of all, requirement is the detection of those particles. And already some experimental setups are there, like underground massive water tanks are being used to catch them. But only a fraction of them are being caught by such huge devices or huge setups. Underground massive water tanks are being used. Anyway, so now scientists are, CRISPR. This is what? This is a revolutionary gene editing tool. And this is famous for editing human DNA. So now scientists are asking, can DNA also be used as a recording device for cosmic events? That is their question. This is a question of the scientists. Can DNA also be used as a recording device for cosmic events? So what is the idea? You can easily understand that this idea is astonishing. But what is the idea? Here we are talking about CRISPR, DNA technology, then again neutrinos are there. So just imagine you have somehow engineered DNA molecules, means you have engineered DNA strands. And when neutrinos pass through them, then in that case some changes occur into the, means you are using what? DNA molecules. So you can expect some changes occurring in the strands. That means actually in the genome itself, some changes are occurring when neutrinos are passing through them. So you need to engineer those DNA strands that way so that they can record that trace. And later, by using CRISPR technology, if you could read or amplify those traces, then you would be able to know the details of the neutrino particles. Okay, so first of all, what is there? DNA strands are there. And when neutrinos, means when those DNA strands are struck by neutrino interactions, when those DNA strands are struck by neutrino interactions, some changes occur. And those changes are what? Those changes means the strands have recorded the cosmic event. That means those neutrino particles, when and where they passed through. Those things are recorded that way. And later, what will be the requirement? Later, CRISPR tool will be the requirement. By using CRISPR tool, you could actually do what? You could read or amplify those traces to know when and where the neutrino particles pass through. And just by scaling up this method, say for example, millions or even billions of engineered DNA molecules you are using, because you need to scale up the same method. That was the idea. Changes in the DNA strand, when those DNA strands are struck by neutrino interactions, and then again by using CRISPR tool, you are reading or amplifying those traces. Okay. So just by using a single DNA strand, our purpose won't be fulfilled. To solve our actual purpose, we need to do what? We need to scale up the same method. And that's why you need to use millions or even billions of same DNA molecule. So this is just like what? Shrinking the entire, that massive underground tank into a test tube filled with engineered DNA. It is equivalent to that, okay? You are using a test tube filled with engineered DNA. This is what? This is your new lab. What was the old lab? That massive water tanks to catch those neutrino particles. That means that underground lab, you are shrinking actually that underground lab into a test tube filled with engineered DNA. So this is what? This is just the idea at present. This is not being used. This technology is not being used at present. Scientists are thinking this way. Okay, they are thinking this way. So you can easily understand, if you have some idea about CRISPR technology, then you can easily understand that yes, this is possible. This is possible. And that's why scientists are planning so. So this was the total idea about that CRISPR and neutrinos. Okay, wonderful intersection of biology, means this new technology is lying at the intersection of biology and physics. Anyway, so this was all about today's topic. If you found this video interesting, so please like, share and subscribe to our channel. This is the end. Thanks for watching.